Well, good afternoon, everybody. I'm going to open and welcome you to the FOODHAY webinar this afternoon. My name is Michelle Williams, and I am the leader of FOODHAY, this activity that you're going to hear about this afternoon, and I'm also the head of Department of Food Science at Aarhus University. I'd like to welcome you today to this webinar and actually to thank you for showing your interest by joining us for the next 2 hours to hear a little bit more about FOODHAY. Actually, we have some 180 people that have registered for the webinar today, and I can see that we are around the 50-odd people. So as happens post-COVID, people just leap onto these online sessions at the very last minute. So we will let them continue to join us, and we will start to run the program. So now we are here. I would like to start the webinar by saying that I'm going to give a small introduction, and then you will hear 7 short presentations about the equipment and how the equipment is being used, and how we are using some of the equipment to solve various research questions. Now, we've got 7 presentations today, and they will showcase various pieces of equipment, but I need to emphasize that within FOODHAY we have a collection of over 100 pieces of equipment, and we do not have 100 talks today. So therefore, what you're hearing today is a small snapshot of some of the equipment and how it's being used to answer research questions. And then I will encourage you very much to look at the FOODHAY website and get to know a little bit more about what we're doing in the universities across Denmark. So as a small introduction, FOODHAY, as you can see, is an open innovation food and health laboratory, and it's a high-tech national distributed laboratory. And what that means is that it's not at one place in Denmark. It is located in Aarhus, and it is located in Copenhagen, and I will explain that in a second. And this collection of equipment and laboratories has state-of-the-art research infrastructure, and the focus of the areas that we are working on is healthier and more sustainable foods. And who are the partners in our, who are the partners in FOODHAY? The research organizations are Aarhus University, Danish Technological Institute, DTI, the Technology Institute of Denmark, DTU, and the University of Copenhagen. And then we have a strategic research partner there, a company, which is Arla Foods. And we also have representation by some of the Danish food companies through a collaboration with DI and Food and Biocluster Denmark. And they sit and talk with us and discuss with us about the use of the equipment. So those are the people that are sitting steering this very large project. And when I say very large project, it's because we applied for money and finances from the Danish Ministry of Higher Education and Sciences. And this is part of the national roadmap for research infrastructure, and we applied for 51.5 million Danish kroners, and that's what we were awarded by the government, by the ministries. And the 4 research institutions and Arla matched the funding, matched the size of the grant, and in total, the grant then, or the project, or FOODHAY, actually is the sum of 103 million Danish kroners, which is an extremely significant investment in research infrastructure within the food space. So how does FOODHAY actually look? If you go onto the website, which you will be explained where that is, then all of the instruments are aligned to 4 main platforms. And these main platforms are food biophysics platform, and the biomarker and screening platform, the sensory and consumer platform, and proteomics and metabolomics platform. And under each of these 4 platforms, there is a whole series of labs, and under each of these labs is a series of equipment. So I'm just going to take you through those very quickly. The food biophysics platform, that is to examine and measure structural properties in foods and contact materials, including packaging materials. And the platform also includes equipment that can be used directly in the production of food. And you can see there that there are a series of labs that are aligned to this platform. And under that, there is equipment that is located in Aarhus or located in Copenhagen. Then we have the biomarker and screening platform, and that has a collection of equipment that identifies profiles, sequences, extracts, ferments, different types of biomarkers, including both pathogenic and health beneficial biomarkers in human, animal samples, plants, as well as in raw materials and in foods. And you can see there that there is quite a selection of labs that are underneath the biomarker and screening platform, and there is a significant amount of equipment purchased to support this type of research. The next platform is proteomics and metabolomics, and proteins, peptides, amino acids, lipids, minerals, vitamins, and other bioactive substances. And this requires very high-end equipment. And you can see there that we have a series of labs that contain some of the equipment that we have purchased under this platform. And then the final fourth platform is sensory and consumer sciences. And this is focused on consumer and sensory research facilities, including product development, sensory profiling, as well as spaces for panel discussions. Oops, that was not me. As well as spaces for panel discussions and determining consumer preferences and behaviors. And we have 2 labs there aligned to that platform. So overall, we've purchased approximately 100 pieces of equipment. And this equipment has then been used to support around 180 collaborations between the research organizations, the FOODHAY partners, and various companies, both nationally and internationally. And also over the last 3 years, 2 to 3 years, we have involved 128 collaborations with various researchers from Danish and international research organizations that have then used this equipment to be able to gain new knowledge or to train new talent. The research has been published in to date, 41 published articles in 21 and 22, and 35 conference procedures. And it has underpinned research from 37 bachelor's and master's theses and 34 bachelor's and master's educational or teaching programs. And these numbers that I have here are from 21 and 22, and we will, within the next 6 weeks, update the website to include the 2023 data. So how do you access this equipment? You access this equipment primarily through collaboration with the research organizations that I mentioned at the beginning of the PowerPoint, because the equipment is designated into one of 3 access levels. The first level is a relatively open sort of laboratory where it is possible for companies or collaborators to come in and use that equipment with what I would say the minimal amount of training or supervision. So we have some open, but because this is about excellence and because this was to increase the competitiveness of the Danish food industry and the Danish food researchers, much of the equipment fits into what we would say excellence or very advanced equipment. We've tried not to duplicate the equipment across Denmark. We've tried to make sure that we have used the funding to lift the whole level of equipment that is available in Denmark. But this equipment is very top end. Therefore, it requires a lot of skills to be able to use the equipment, and you will hear from some of those very skilled people today as to how they have accessed the equipment, how they have used the equipment to solve specific questions and to gain new knowledge. And therefore, it requires quite a lot of expertise and training, not only to use the equipment, but to understand the data and the results that come out from using the equipment. So the best thing to do really is to look at the website and to find the various pieces of equipment and how we are using that equipment. And every piece of equipment has a contact person, or you can look at the videos and you can contact the people that are on the videos, and you can reach out to the collaborators and work out how the equipment can be used to help you solve the questions that you have. So I'm going to actually stop there, because I know that with webinars, they often drift. So therefore, I'm going to stop with my presentation, lift down my presentation. I'm going to get to the next speaker, Glenn, to put up his presentation so that we can try and make a little bit of time here at the beginning of the webinar. So I will ask Glenn.
Yes, hello.
Hello.
Good afternoon, everyone. Just
Good
set it up here.
afternoon.
Yes. So, yes, thank you for letting me present here today. I will present on fundamental. My name is Glenn, and I'm a PhD student at the Department of Food Science here at Aarhus University. And I work within the group called Food Quality Perception and Society. And I'm presenting these things that has been made within our group, and also some of the things that I have made myself. So the presentation is on fundamental sensory interactions and their implications for healthy, sustainable, and acceptable new green product development. So I will start by presenting some of the equipment that has been funded by the FOODHAY, because I will introduce several pieces of equipment. So the first one I want to present are these convertible tables that we use in our discussion room with our sensory panel. So these booths are convertible, so we can make them into a booth. That is very useful, because when we're discussing and are serving some samples, we might want to have screened the panelists so they're not interacting. And the other one I want to present to you is our professional ovens that is funded by FOODHAY in our preparation kitchen. So we use this to heat food and prepare food in several ways for sensory analysis. And the last thing in this round is the extruder I want to present, and this is located in our process hall. This is a very big piece of equipment that can take meals or powders and turn them into extruded products with their various textures. So I want to present some of the projects that has used some of these equipment here. The first one is a project called Meat Analogs. There's a research project on textures, investigating textures of plant-based meat analogs that are based on different plant-based protein sources. And this relates to the valorization of side stream products. That could be like leftover meal or cake after oil extraction. It could be, in this example, it's hemp seed. So we want to make these side stream products into new green products. And we can do this with this extruder, where we can make highly fibrous whole cut meat analogs using less refined hemp protein concentrates, or that's some of the projects have investigated if we can use these less refined sources. So the concept that we take these powders, protein powders, extrude them into different textures. Then we can cook these products, and we can actually make sensory analysis on them to find out the textural differences of these and compare them to real meat products. And we have made several publications now on these types of extruded proteins. This is one that was just published yesterday, where we have uncooked and cooked extruded, where we actually did a sensory analysis of these extrudeds. And the same with pumpkin here. So that was a short show off of this project. Now I'll present another project called Waste Sense. So this relates to the topic of reducing food waste, and Waste Sense is around like evaluation of freshness in meat, and this was about developing a high precision sensor that could predict the shelf life of foods, of meats and fish products, to actually reduce food waste. And that included that we had to like validate the cadaverine which this sensor measured as a spoilage indicator in these food products. And then also we wanted to calibrate so we can predict meat and fish shelf life based on these cadaverine sensor measurements. And in the end, we did freshness evaluations to further validate the freshness of these products using sensory analysis of the food products during storage. So this was primarily where we used the convertible tables, and we just received this FOODHAY funded thermal chamber where we can actually control shelf life parameters for in future studies. And now I want you to show you a very different project, the things that I'm working on, and this is where I'm using a 5-channel gustometer in our observation lab. So this is just a pump system, a high precision pump system that can deliver solutions to the tongue of the participants that is actually just normal consumers I invite for the studies, and they will evaluate the taste solutions that I spray onto their tongues. And it has a split tongue design. You can see there's a small white plate here, so I can actually separate the taste solutions that are sprayed onto the participants' tongues. And this is something I exploit in this research. So application of this gustometer, and this is just a bit of a general introduction to it, because it's not easy to understand this method that I'm using here, this split tongue method. It took me a lot of time looking at the results many times to actually just understand these results I got out of this equipment here. So what I want to do is that, or what we want to do, we have had several projects now actually using the gustometer with the split tongue setup, is that we want to understand fundamental taste interactions. So it's interactions between the sweet, bitter, sour, umami, and salty tastes. And we're only doing that by spraying these solutions on the participant's tongue and having them evaluate how they perceive these. So we know that all of these tastes can actually influence each other. They can enhance each other and they can suppress each other, which is very dependent on the concentration, of course, which we also vary. But we all know that when we combine, for example, a bitter tasting coffee with some sugar, we can decrease the bitterness of this coffee. And that is very variable what happens when we combine these different tastes. So we want to understand these fundamental taste interactions to actually facilitate like healthy, and sustainable, and acceptable new product development. So a bit on this split tongue setup that we're actually using. So what we can do is that we can look at, we want to look if these interactions that are happening between the tastes, if they are central interactions happening in sites in the brain or in the afferent nerves, or is it something that happens on the tongue? And we can do this by either sending the different compounds individually on each side, separately on each side of the tongue, or we can combine them on one side of the tongue, and then we can look at taste intensity differences. So what I've been looking on is sweetener interactions. So does sweeteners taste sweeter together? And what happens when we actually mix these or separate these? So sweeteners, they can synergize, and when they do that, then it can reduce off taste, and it can lower cost or provide ingredient functionality that is actually often lacking in these individual intense sweeteners or non-nutritive sweeteners. And sweetness synergy in itself is something that happens when we mix these sweeteners. So it's the effect of the mixture of these two sweeteners. If that is greater than would have been expected from each in isolation, then they show sweetness synergy. And what I'm focusing on specifically is stevia, and comparing them with other synthetic or artificial sweeteners. So does stevia actually blend well with sugar? And that is what I'm investigating. Also because stevia is a natural sweetener that consumers demand, and it's also able to actually reduce has a lower carbon footprint than sugar. So here's a sucrose control, where I actually show that there's not a difference between presenting these. There's no interaction happening when I'm presenting sucrose separated or combined. So this was my control. This is additivity of sweetness. Then I have a synergistic control. I can see that when I combine them together, they show actually that there's some peripheral interaction on the tongue where they synergize. The same I can see when I combine sugar and stevia. They actually have a higher sweetness intensity. This is the synergistic control is a known pair of sweeteners that actually synergize. Then also bitterness suppression. Is that also peripheral or central interaction? And can remove the bitterness of the stevia? This is the peripheral control. We saw that the bitterness of one of the sweeteners could actually be suppressed when we mixed them, but not when they were separated. So that showed us like that we were able to reduce the sweetness with these, and this is confirmed in cell studies have been earlier. Whereas I could not do this with stevia, unfortunately. So the last thing, our future study I want to show you is that what we are planning to investigate is kokumi taste interaction. We want to understand these interactions happening between kokumi, this newly found or very investigated flavor compound or flavor modifying compound, and its interaction with umami, because it can it's a way to improve the formulation of these plant-based foods that we see a lot. So kokumi are these tripeptides that have multi flavor modifying properties. They have very little flavor on their own, so it's very interesting what they can actually do, because they can induce or enhance thickness or mouthfulness of different taste compounds. So it's actually shown that it's interacts with the calcium sensing receptor. And also that it can enhance the mouthfulness of sweet, salty, and umami. Oh, sorry. And this is actually what we want to investigate, if some of these interactions are peripheral taste interactions on the tongue, or if is it something that happens in our brain in central interactions. So that was it for me now. I know that I've used more than I should, but that was all from me.
That's very good. That's very good. Only slightly. So you're good, Glenn.
Yeah.
Well, what we will do then is we will move on swiftly then to Stig. And thank you, Glenn, for that. Stig, would you like to upload your presentation for us?
My name is Stig Skrivergaard. I hope you can all hear me nice and clearly. I'm currently a postdoc at AU Food, and last year I finished my PhD in cultivated meat. And yeah, now I will present some of the stuff that has been enabled by the FOODHAY funding. So just briefly, I'll just not mention like the entire thing about cultivated meat, but for those who haven't heard, it's about taking these stem cells from animal tissue, and then the idea is to cultivate them and expand them in large bioreactors, and then in the end form essentially muscle tissue, or we'll get the cells to create myofibers on a three-dimensional scaffold in the end, and then replicate sort of that meat texture with actual muscle cells. So what we mainly use in the lab is these satellite cells. So it's essentially a muscle stem cell. It can proliferate, so it can do a lot of cell doublings, and then it can also differentiate and fuse into these long multinucleated myotubes, which is an early state muscle fiber. So first up, we also got a new cell lab because of FOODHAY funding. So that's quite amazing, because we essentially doubled our working space now with 2 extra flow benches and 2 extra CO2 incubators and a cell counter in this new lab. So plenty of space now for all the projects that are ongoing with cultivated meat and cell research in general. So that's really great. And now I'll present the Citation 5 equipment, which we received during my PhD, and I've been working with it quite a lot, and it's been a really, really, really useful machine for cell research. So what it basically does is that it can take pictures, so it's a fluorescent microscope, and it can do this in a high throughput format, and we can then analyze the cells in the proliferative stage, but also in the differentiation state. So one assay is just staining the nuclei of the cells with a color called Höchst. So it's cell permeable. It's really, really fast. It just permeabilizes the cells and stain the DNA inside of the cell. And here we can really robustly screen a lot of different compounds in terms of proliferation. So we basically just count the cells in a high throughput format. And what we had before getting the machine, then we did something. So it was basically a metabolic assay. So we measured the color change or absorbance change in the media just with a normal absorbance plate reader. And this is a lot of samples here with a lot of different compounds in them. There's no change here. So the same samples here were just counted cells with the Höchst stain, much more sensitive. So we can actually see small differences between the samples, and that really enables us to optimize different aspects. For example, a serum-free media that I developed in the lab. So really great for this, but it can also do more difficult samples. So for example, these differentiation parts in the cells. So here you can see these long myotubes, here colored green with a stain against myosin heavy chain, one of the functional proteins in muscle contraction. And here the software then recognizes based on this intensity of the color, and we can then quantify the amount of myotubes actually being formed in the samples. And in a similar case, here it's a stain against the actin filaments, a stain called phalloidin. And it basically does the same trick. So here we just stain actin, which is very dense in these myofibers. So basically early muscle fibers. And here we can also very precisely quantify the amount of myotubes being formed, but also with the Höchst staining added, we can then also quantify the amount of cells totally in the sample, but also how many nuclei that are within these muscle fibers. So we can calculate something called the fusion index. So the amount of cells in the sample that are actually going into these muscle fibers. And that's also a very important parameter that we are looking for in the samples. And then we can basically put in our plates, and we can cultivate the cells inside of the machine and take regularly images of this, and then we can make beautiful videos like this where you can see them grow and form muscle fibers. We can do that because we can attach a CO2 gas to the machine as well. So it actually works as an incubator as well. But what is more cool than this, to me at least, is that we can also quantify it through something called high contrast bright field imaging. So this enables label-free counting of the cells. So the other one, Höchst, and the staining against actin, there you basically kill the cells and you fix them. But here we can continuously analyze the samples throughout time through something where we defocus the image, and then we create a contrast basically and let the software again identify based on contrast. We can do this for multiple days and get really great high resolution dynamic growth curves like this. So this is really, really useful when we're testing different components in the media. The same technique I've actually developed sort of this novel technique where we can use the same principle and then actually quantify the amount of myotubes as well in a label-free setting. So it's the same principle. Again, it's a defocused image that we pre-process, and we have a lot of contrast then. We can quantify the myotubes in this sample, and then in the same manner, we can put in the plate and we can just quantify these being formed in real time over several days. And with that, we can screen, again, different components in this very dynamic assay and look at differentiation, which is much more complex than the proliferation itself. Yes, so the Citation 5 is a really, really powerful image-based cytometry equipment, and it's been, indispensable in the cell lab, and it's being used daily for the proliferation assays and differentiation assays that we're doing. We are also using it for other types of assays. We can also quantify the amount of lipid droplets that can accumulate inside of the cells when we add lipids to the media, and a bunch of other assays. So this basically no limit to the amount of different assays you can set up, if you can sort of imagine it, and there's a specific dye for what you're looking for, then we can basically quantify it, that very robustly in a high throughput format. Yeah, and I would just mention that I used this specifically for 2 papers I published last year. It wouldn't have been possible without this machine, so I'm really grateful. So the left one is where I designed the serum-free media. You can see some design of experiment graphs and some response surface methodology where I optimized the concentrations of some serum-free components, and that was using the Höchst based assay, so very robust and very precise assay. And then to the right, this other paper where I compared satellite cells isolated from younger bull calves, compared them with older dairy cow samples. And here I used the label-free quantification as well as the fusion assays with actin and myosin heavy chain staining. So yes, we're really grateful for both the Cell Lab and this super great equipment that we have here. Yes, that was me. I hope I went through.
Yeah, that was very good, Stig. Thank you very much for that introduction. Of course, there are so many questions related to the cellular meat space, how to develop meat from cells, and where to source the cells from, and how to do this in a sustainable way. So many of the questions that you are looking at in your research are fundamental questions that we need to know to move from sort of more traditional animal-based products to more sustainable but still animal-based products, which is important because a lot of people still want to eat animal-based foods and source protein from them. So you're really doing some great research there. I think we will move forward then. The next presentation actually is also from DTU, and one of the speakers is not able to join us today. So therefore, Charlotte Jacobsen is actually going to be kind enough to present and give us an insight into their research around both cocoa products and around microalgae pigments. So I will hand over to you, Charlotte.
Great, thank you. So I'm just going to minimize over here. So yeah, thank you so much. And so what I will be presenting today is the UHPLC-MS equipment we got from FOODHAY to particularly work on antioxidants and pigments. And before we got the equipment, we had been using HPLC also for the same type of analysis. But of course, by having a UPLC-MS instead of a HPLC, we were both able to perform the analysis faster. And we were also able to do the identification, a more secure, if you can put it that way, or certain identification of the compounds by the mass spectrometry. So today I will be giving examples from both how we have used the equipment for antioxidants, but also for pigments. In general, when we were applied for the equipment, it was for testing oils and extracts from algae and food emulsions, but we also had extracts of other foods in mind. And that's actually one of the examples that I will show you here today is from cocoa beans. So we are actually starting with that. So this part of the presentation that my colleague Ditte was supposed to have presented. But I will take over and do my best to convey the messages that she wanted to convey. So what Ditte was doing in this project was to look at the processing of cocoa and how what that would do to antioxidants, particularly phenolic compounds. So the first slide here it illustrates the general work process that we have, no matter what compound that we are looking for. That first we will run some samples, and then we will select some of the compounds that we want to look further into by using the facility to the MS facility to identify the most interesting compounds. And then we will, after that tentative identification, we will get the identification confirmed by buying some standards that should hopefully fit with what we have identified by the MS. And this is exactly what we did here. So we found that the most prominent flavonoids in cocoa was those four listed here: catechin, procyanidin, epicatechin, and epicatechin gallate, ECG, and then also theobromine. And theobromine is actually one of the compounds that give bitterness to the chocolate. And we were working together in this project. We were working together with Mellow Chocolate. And Mellow Chocolate had this idea that they wanted to have as much of the positive phenolic compound, you could say, as possible, and at the same time minimize the content of theobromine. And the content of these phenolic compounds is actually changing during the processing. And here on this slide, you see the processing of the cocoa bean. So you have and all the way actually to the chocolate. So during this processing, particularly during fermentation, drying, but even more during roasting, you have a change in the composition of the phenolic compounds. And what we knew prior to the project was that especially this epicatechin would change when you heat to more than 150 degrees, then it will epimerize into catechin. So that was one of the things that we would be looking at during this project. And what we were actually working with different cocoa beans, and here I'm showing one example of what happened during the roasting process, because that's where we put most of the emphasis, as I mentioned before. So here we have it is a Chuno Clásico from Nicaragua that we were then roasting at 2 different temperatures for different times. And you can see that, as expected, that when we increased the temperature to 150 degrees, we got more of the catechin, and you would expect maybe that we would get less of the epicatechin, and we also did that, but it didn't happen until after 15 minutes. But that's also where we really saw the increase in catechin, so that fitted quite well. We also looked at the theobromine, as you can see on this slide, and here we could see that particularly after 15 minutes, we started to decrease even faster. So that would be the parameters that you would play with here, both the roasting temperature and also the time. But we could also see that when we were testing different beans, that the profile that you would get and also how the profile would change with time and temperature would actually vary from bean to bean. So from this project, we have communicated the results at several occasions. We've made an oral presentation at Eurofed Lipid and also at IDA, and we have also submitted a manuscript, and hopefully that will soon be out, so you can read more about the results in that manuscript. If you are interested to hear more, you're always welcome to contact DIDA, of course. Then the next case that I will explain a bit more about is some work we did on lutein from microalgae. And here again, we started with the same process that first we identified what was the compounds that we had present in this microalgae, and what was the and then we used this. We got the standards, and then we were quantifying. So you have the calibration curve here to the right, yeah, you can see that I have listed here lutein, fucoxanthin, chlorophyll A and B, but you can actually see in this chromatogram, you can see several other compounds that we were also able to identify, and which we would also have been able to quantify. But here in this project, we were concentrating on these 4 compounds. And this project that I'm showing you here was actually in collaboration with the microalgae producer Liga, and they were interested in developing a method for the extraction of lutein. And lutein is a yellow-orange pigment that is currently, or that is now, you can get it from another plant, but it's quite expensive. So we thought of that we could maybe find alternative sources, and that the microalgae could be one of them. So in this project, we tested various different methods for extracting the lutein, and it was the intention that this should, of course, happen in larger scale, not only for analytical purposes, but in a process that could then be implemented if it was economically feasible. So what you see in the top here is the microalgae powder before extraction, and then this is actually the extract that we obtained, and this is then the residual biomass. And you can definitely see, no matter here how we extracted it, that the biomass got a bit lighter. But what you also see is that the extract that we got was not really yellow, but it was green. And that is because, unfortunately, with these processes, we got co-extraction of chlorophyll, and you see that in the graph here as well. So we have the chlorophyll B is the purple one here, and the lutein is the green one. And then you also see we get plenty of fucoxanthin, and that was actually no matter which kind of pre-treatment we use, because we also tested different pre-treatments. We got the chlorophyll out as well, and this chlorophyll is so green that it masks the yellow color of the lutein. So then we were looking into whether we could further purify our extracts, and one of the methods that we looked into was to first do this supercritical CO2 extraction of the lutein, and then afterwards do column chromatography. And now you see we got a nice and yellow extract, and actually the recovery of the lutein was pretty good, but still, when doing the calculations, the company did the calculations that even though we had a successful extraction of lutein, but the current market price of lutein, even though it's quite expensive, this technology is still too expensive for them to be able to implement it and have a good outcome economically of that. So that was, and I forgot to mention here, yeah, that we have a manuscript in preparation. So soon you will also be able to read more about those results. And apart from that, we are working with the equipment for other projects, both with microalgae, but also with seaweed. So it has been very useful for us to get the equipment in the lab, and again, we would also like to thank FOODHAY for providing this opportunity. So thank you.
Well done, Charlotte. That just shows that the power of science, that when we think that we can extract something, it's not always as simple as we think it's going to be.
Yes.
And also just how dominant chlorophyll is as a product, the fact that it's protecting itself out there, which is actually also important for us. But Our next speaker is from the Department of Food Science at the University of Copenhagen, and we'd like to hear from Lichuang today, and we look forward to hearing a little bit more about your research. So thank you very much.
Good afternoon, everyone. I'm Lichuang from the Department of Food Science, University of Copenhagen. Today my topic about the quantification of kokumi peptides using LC-MS. I will begin by providing the background information. Gamma-glutamyl peptides, also known as kokumi peptides, have attracted increasing attention due to their kokumi sensation and taste-enhancing ability, which can intensify the mouthfulness, thickness, and the continuity of foods. Furthermore, they can also strengthen the intensity of basic tastes, such as sweetness, saltiness, and umami. Gamma-glutamyl peptides have been mainly identified in plant proteins and fermented foods, including fermented fish, cheese, soy sauce, yeast extract, and beans. Gamma-glutamyl peptides are mainly produced through enzymatic reaction catalyzed by gamma-glutamyl transferase. For the enzymatic process involves hydrolysis reaction and transpeptidase reaction. Firstly, the hydroxyl oxygen on the active site of gamma-glutamyl transpeptidase will attack the amide carbon on the donor substrate, generating gamma-glutamyl enzyme intermediate. When water molecule act as the acceptor substrate, generating glutamate. When gamma-glutamyl containing amino acids or peptides act as the donor substrates and acceptor substrates, leading to the occurrence of auto-transcriptation. For the assessment of the transcriptase activity, there are mainly two methods. The first method is using a synthetic substrate, gamma-gpin, act as the donor substrate, D-glycine act as the acceptor substrate. The resulting yellow compounds PNA released during the reaction could be detected by a plate radar or spectrophotometer at 410 nanometer. For the second method, when glutamine or gamma-glutamyl containing peptides act as the donor substrates, generating gamma-glutamyl peptides. The gamma-glutamyl peptides could be detected and codified using LC-MS. Moving on to the quantification of gamma-glutamyl peptides. The distinguishing between the alpha-glutamyl peptides and the gamma-glutamyl peptides pose a challenge due to their similar structure and identical molecular weight of the alpha-glutamyl peptides and gamma-glutamyl peptides. As shown in Figure 1, take alpha-glutamyl tyrosine and gamma-glutamyl tyrosine as an example. The protonated alpha-glutamyl tyrosine show a loss of water molecule and the formation of iron 102. Whereas for the protonated gamma-glutamyl tyrosine show a loss of ammonia and the formation of iron 130. This iron was selected for the quantitation of the gamma-glutamyl tyrosine, and alpha-glutamyl tyrosine didn't contain the iron 130. But the gamma-glutamyl tyrosine contains this iron. This iron was used to identify the gamma-glutamyl peptides. Utilizing the OpTrap explorers LC-MS, the parallel reaction monitoring technique was used to identify and codify the gamma-glutamyl peptides. For instance, this is a mass spectra of the protonated gamma-glutamyl tyrosine detected by the OpTrap explorers LC-MS. As shown in this figure, we can find that the parent error and this is the specific error, and we can also find the error 130. Table 1 shows the mass charge ratio of the parent and fragment error along with the retention time of the 13 different gamma-glutamyl peptides. For the fragment error and the retention time, we use it to verify the presence of the corresponding gamma-glutamyl peptides. And the specific error of the target gamma-glutamyl peptides, we use it to codify the concentration of the gamma-glutamyl peptides based on the standard curves. Let's move on to the application. The application one is focused on the identification and activity characterization of the bovine milk gamma-glutamyl transferase. Firstly, the enzyme responsible for the transpeptidase activity in bovine milk was identified using LC-MS based on the in solution and in gel trypsin digestion methods. Afterwards, gamma-glutamyl peptides was generated by bovine milk gamma-glutamyl transferase. And the reaction product gamma-glutamyl peptides was identified and codified using LC-MS. This study reveals that the bovine milk gamma-glutamyl transferase could synthesize different type of gamma-glutamyl peptides. Application one investigates the substrate selectivity and catalytic efficiency of the gamma-glutamyl transferase. As shown in this figure, this shows the gamma-glutamyl peptide concentration generated by bovine milk gamma-glutamyl transferase. We can find that the bovine milk gamma-glutamyl transferase has the highest affinity with well-glide acceptor. This gamma-glutamyl peptides have the highest kokumi intensity. And as mentioned earlier, when we assess the transpeptidase activity, we use the gamma-glutamine as the donor substrate. And when the glycine and the glutamic act as the acceptor substrate, both commercial gamma-glutamyl transferase and the bovine milk gamma-glutamyl transferase did not show any transpeptidase activity. But when the glutamine acts as the donor substrate, the corresponding gamma-glutamyl peptides, the gamma-glutamyl glycine and gamma-glutamyl glutamate generated for both the commercial and the bovine milk gamma-glutamyl transferase. Especially for the concentration of the gamma-glu glycine, it has the highest yields. This study reveals that the gamma-glutamyl donor and acceptor substrate could significantly affect the catalytic efficiency of the gamma-glutamyl transferase. Application 3 mainly focus on the optimization of the gamma-glu well-glide production by optimizing the pH, substrate, sodium chloride, and the enzyme concentrations. The resulting gamma-glutamyl peptides will be detected by our TSQ, LC-MS. For TSQ, LC-MS, the selected reaction monitoring was used to detect and codify the corresponding gamma-glutamyl peptides. For the SRM technicals, we need to select the which error was used to codify manually, but it can't detect the fragment error derived from the target gamma-glutamyl peptides. The TSQ LC-MS is suitable to detect kokumi peptides in simple systems, but for the complex systems, they may contain some alpha-glutamyl peptides and complex matrix. So it may affect the codification of the gamma-glutamyl peptides. In this study, we obtained a relatively high yields of the gamma-glu well-glide. That's it. Thank you.
But we can move to our final presentation today. Today we also have a presentation around emulsion stability from Anne-Louise, and she is at a DTI. So I can see that Anne-Louise, we can hear you, I believe.
Yeah.
Yes, we can hear you.
So.
So that's very fine.
Perfect. Yeah. Hi, I'm Anna from Danish Technological Institute, and I will present a little bit about two of the equipments that we have here. The first one is a wet mill, and the second one is a turboscan, which can assess the stability of opaque samples. And I will use a plant-based yogurt as a case study. But first of all, a little bit about the equipment that we have been using in this study. It's a Dynamill multilab wet mill, which contains beads. You put in some ceramic beads with different diameters in the chamber, and if we look into the machine, it has this shaft which is rotating, and this rotating discs, which makes the beads go around in the chamber, and then you have a liquid sample with some particles in. You pass through, and when the particles hit some of the beads, they will break down into smaller pieces. So this wet mill can be used for particle size reduction and to improve the appearance of, for example, brightness of colors, and it can increase surface of some of the milled product, and it can also reduce sedimentation. And what we have used the equipment for in this study is particularly particle size reduction and reduction of sedimentation. To assess the yogurts we got out from the plant-based that we produced on the wet mill, which I will tell a little bit more about in a minute, we used this turboscan tower, which use static multiple light scattering as measuring principle. And as I mentioned, it's used to measure the stability in opaque samples, which a yogurt is. And the measuring figure, which is used for the equipment, is called turboscan stability index. And if you have a turboscan stability index above 3, you would be able to see the destabilization of your sample with your eyes. And a benefit of using this equipment is that it divides your sample into 3 sections, so you can assess the top section, the middle section, and the bottom section. As a result, you get out a TSI of the global sample, and if you see destabilization, you are able to dig into the sample and see if the destabilization is more pronounced in the top, middle, or bottom of your sample, to see where you have this sedimentation or flocculation, or what is causing the destabilization of the sample. Another advantage is that you can have a temperature range, so you can measure from 4 to 80 degrees, where you can mimic shelf life in a refrigerator, in our case for a yogurt, but you can also do stress testing by increasing the temperature. And then it's possible to have 6 different samples at a time, and you can measure over a period of long time. There's multiple options for setting up the precise measurement that you desire. And now for the case study, which was plant-based yogurt, and the aim of the study was to produce plant-based yogurts on Nordic protein sources such as pea and faba bean. And one of the things we looked into was how we could make a stable plant base that we could ferment on, and how we could produce a yogurt without adding stabilizers and still having it to be stable at the end. So one of the first things we did was just to make the plant base by mixing it, and what we saw was this large sedimentation here. So we quickly realized that we need some kind of better suspension of the proteins in the plant base before we could ferment on it to achieve the best fermentation. So we realized that we have this wet mill available here at DTI, and we thought since we already had our proteins in a liquid medium, we looked into wet milling as a solution. And what we found out is this is just the plant base here, wet milled 5 times. And as you can see here, the sedimentation is much less, but it seems like there's still some slight sedimentation here. So we found out that wet milling was a good solution, but we also found out that heat treatment prior to wet milling and then wet milling was the best solution, because as you can see here on the last picture, there's no visual sedimentation. And as an additional benefit of this wet milling, we achieved a reduction of particle size, which you can see here. The red or the orange line is a sample which has only been mixed, so it is this sample down here, and the other one, the blue line, is a sample which has been wet milled. So you can clearly see that the particle sizes decreases after wet milling, and in fact that was also what we saw when we did some sensory assessment of the sample prior to wet milling and after wet milling. And prior to wet milling we had this grainy mouthfeel, and after wet milling it disappeared. So actually we've gained this additional benefit, and we had some plant bases that we were happy with, and we thought that the stability was good enough, so we could proceed on and doing some fermentation on the samples, and we produced some plant-based yogurts where we assessed the stability of 2 of those and benchmarked it against 2 commercial products, a dairy product and a plant-based product based on soy. And as you can see here, as I mentioned before, the TSI, if the TSI is above 3, you can see the destabilization with your eyes. And for us, this is a good result because all of the samples are below 3. And in fact, the faba bean yogurt was the one who performed best at a period of over 7 days. And this is performed at 4 degrees. And we've in fact also did a test at 20 degrees over a period of 7 days, where we also saw good stability also compared to these 2 commercial products. So from this TurbiScan, the stability assessment, we can conclude that we achieved good stability over time, and this is without adding any stabilizers. As you can, when you read the ingredient list in the, for example, the Alpro yogurt, which is the plant-based yogurt, already commercial, they use stabilizers to make their product stable over time. And we achieved the stability by just fermenting on the plant basis. So all in all, we found out that heat treatment and combining it with wet milling gave us a good suspension and good emulsion, and gave us smaller particles, which were very desirable, and that we could produce and assess the stability of these plant-based yogurts, and they were good over time compared with the commercial products. So all in all, we succeeded, and as you can see here, we have 3 examples of the yogurts that we managed to produce on these Nordic plant proteins as ingredients. And there's also some, I will just highlight some other possibilities with this wet mill, because it's capable of doing multiple things, and we use it for multiple things in our daily work. For example, a most common technique for milling today is dry milling, because it's easy and don't take a lot. But sometimes you can have a raw material that can't where dry milling is not suitable. For example, this could be a product with a high fat content, where the friction made in this dry milling just melts the sample with the high fat content. Here, wet milling could be a good solution, because you also have a cooling jacket here, so the temperature is kept down. And of course, you need to have a product which can be suspended in liquid, and this could be both water, but it could also be oil or other things, but it has to be suspendable. And furthermore, we have also used it for particle size reduction, as I also mentioned, which is great because it makes it easier for our product developers to incorporate some ingredients which are normally found in larger particles into application where they can kind of hide the graininess with these larger or smaller particles. And it could also be where the dry milling is not suited. It could also be some ingredients which are sensitive to pH or pressure, or to temperature or pressure or other things like that. Here, the wet mill is a good direction to look into if you have such problems. And as a final remark, I will just briefly show some of the related FOODHAY equipment that we have available here at DTI, which are capable of making homogenization and also emulsification. At the left, we have a small lab scale homogenizer, and at the right, we have high shear Silverson mixer, which can be used for emulsification and homogenization and dissolving and disintegrating of particles. And one of the good things about both the wet mill and the homogenizer and the Silverson is that it's scalable equipment. So the results that we gather here in our facilities are scalable to industries, and that are what many of our customers are interested in, because we work a lot with the small companies, which are startups, and they just need to, sometimes it's proof of concept they need with this equipment that we use. And other times, yeah, they need to have to know that it's scalable. And most of the processing equipment we have acquired through FOODHAY is scalable, which is quite nice for our customers and ourselves. And I think that was it from my side.
I would like to take a second to say that it's really been a pleasure for me to, and I've certainly learned some things today from listening to these diverse talks. It's really been a pleasure for me to have these speakers present some of their equipment and the application of the equipment. And I would like to thank them for the discipline that they've shown this afternoon and their very, very professional presentations, and that they've given us an insight into how they're using just some of the equipment to solve some of their research challenges. And I heard quite clearly that this has been able to, some of these investments have been enabled them to get new results, or a different approach to their results, or more efficiency in the results that they have been looking for. So I think that's really a success overall for the whole partnership. I'd like to thank you as participants for joining this afternoon and listening to this FOODHAY webinar, and I hope that you have found the talks to be inspiring today. I would encourage you to feel free to follow up with the specific presenters that you've heard from this afternoon, but also just to remember that the equipment that we've heard in these 7 talks is a tiny snapshot of the 100 pieces of equipment that we have actually purchased within this very big Danish consortium. So I'd encourage you to go onto the website, because there are contact information of people in all of the research partners across Denmark that have been part of FOODHAY, and where this equipment is located in Denmark. And then you can approach them about possible collaborations. So with that, I know that we are running 5 minutes ahead, but nobody loves more than 5 minutes of free time when the sun is shining, especially in February. So I'm going to officially close the webinar and thank everybody today for participating. So thank you very much, and goodbye from here.